So next, I want to talk about pyruvate dehydrogenase, which is the next step in this energy making process. So far, we've covered anaerobic respiration. This will be the first step that only organisms, or this step that we're going to be covering now is going to be the first step that only organisms that use oxygen will undergo, because other organisms will do something else with pyruvate, which we don't really have to mention. Pyruvate is the product of the glycolysis, and it cannot directly be used in the Krebs cycle. Therefore, it must be made into a molecule that can directly be used in the Krebs cycle, called Acetyl Coa. Okay, and CoA stands for coenzyme A, and it's a really big, complicated molecule that you don't have to know what it is, or what it looks like, but it's just know that it's really big, and it's called CoA. Pyruvate dehydrogenase is an enzyme that is responsible for this conversion of pyruvate to acetyl CoA. It's a very complex enzyme with multiple subunits. I'm sure if you took a biochemistry class, you would have learned about it. It has multiple subunits, E1, E2, E3, and they all have very unique functions that you would have learned about, but you don't really have to know that for this exam, for the MCAT. Know that it is only found inside the mitochondria, and so the first step of the pyruvate dehydrogenase and this process is to transport the pyruvate into the mitochondria, right? You have to get across, you have to go into the matrix of the mitochondria, so you have to get past the membranes. Okay, so this is what the overall formula looks like of the conversion of pyruvate to acetyl CoA. As you can see, there's a lot of stuff that has to go in. The actual CoA attached to a sulfur group, NAD+, which we're converting to NADH. So this is how we make energy off this step. And then TPP, lipoate, and FAD, which are other cofactors. And ultimately, you get acetyl CoA, right, which looks like this, which has this formula, and carbon dioxide. The conversion uses 5 cofactors, which are these 3, and however, you only really need to know about these 2, the CoA and the NAD+. Again, NAD+ important because we actually reduce NAD+ into NADH, and that's how we make energy off this step, because it's equivalent to 2.5 ATP each. And CoA is the actual how we get the coenzyme onto the acetyl group, right? And that's why it's called acetyl CoA. I remember each glycolysis produces 2 pyruvates. So you actually, whenever you go into dehydrogenase, from 1 glucose, you actually get 2 pyruvate. So we actually, most of the time we talk about per glucose. This whole process we talk about per glucose. So we use 2, we consider this dehydrogenase process twice. Okay. So we're actually producing 2 NADH. Okay. So we're producing 2 NADH, and that's how we make our energy off this step. And then, yeah, we have to use 2 CoA enzymes and stuff like that. So I want to move on to now the Krebs cycle. Okay. So this is kind of like the general overview of the Krebs cycle. You start off, you can see, because it is a circle, you can technically start anywhere, but where you actually really start is here with the introduction of acetyl CoA, which you just made in the last step, enters this cycle right here, and then we go this way, clockwise. Okay, so this is a very, very simplified kind of diagram, and I really like this diagram in particular because it really touches on very basics that you'll need to know, right? So you can see here in these steps, the NADH is coming out, right? This is because you're producing NADH. And you'll see in just throughout the Krebs cycle that the main way you get ATP is not by directly making ATP. So in this one step, you are directly making ATP, but it's just this one step, whereas these 3 steps make NADH. This step makes FADH2, which is also similar, right? And the reason is because, again, they're actually worth more. And 1 NADH is worth 2.5 ATP. 1 FADH, and I'll cover this in a minute, is worth 1.5 ATP, right? These are actually worth more, so it's more efficient to make them. And then feed them into the electron transport chain, and ATP synthase, which we'll cover next week. But that's how you actually make ATP rather than just directly taking a phosphate from one thing and putting it on another thing. You can actually get more out of just doing this instead. And then making the ATP kind of using the synthase. Okay. And here you can see where you guys make CO2, where it comes out, right? And that's how you the CO2 you make you exhale, obviously. The NADH is we make energy, FADH I covered as well, and you make GTP or ATP, they're the same thing pretty much, right, in this one step. And honestly, the real Krebs cycle is infinitely more complicated than this. There's the enzymes, they have like a bunch of different stuff going in and out, and like this is not an isolated system. So there's some of these are actually used for other stuff. So like some of these intermediates like Acetyl Coa, succinate, they're actually used for other things like amino acid synthesis and things like that, but that's like way too complicated, right? And I don't want to like show you guys a super diagram, right? So this is just like the simple basic stuff that you probably should know. Sorry about that. Okay, so just like before, when I go over the steps, I'll just be covering what I actually think is like the important part of each step. I would say each step mostly has like one or two like unique things about it that you probably should know and like you should kind of like memorize a little bit. The likelihood that they'll ask you is not very high, but in case they do, like you'll have an idea of it. And this will get you a good idea of what the Krebs cycle is, and like all of the steps of it, without having to memorize all of the enzymes, all of the intermediates, all of like that stuff, right? Like you probably did for biochem. Okay, so firstly, oxaloacetate and acetyl CoA react together to form citrate. So you need both of these together, and you release H2O and coenzyme A, and you then recycle coenzyme A because it's not efficient to make it every time you want to go through the Krebs cycle by converting pyruvate to acetyl CoA. So we recycle it, for instance, it's not used up in this. And notice that oxaloacetate is the final product, but it's also the first product, I guess, right? Or the first reactant, because it's a cycle, goes in here, but you actually need both of these, right? Without oxaloacetate, acetyl CoA can't do anything. And until you put acetyl CoA from the pyruvate dehydrogenase step in, this oxaloacetate will never react, okay? So you actually need both of these together. And this is also a little more complicated, but there is like enzymes that actually regulate this, right? And specific like binding, like ES bind oxaloacetate first and has very specific binding that increases binding for acetyl CoA. And this is one of the ways that you regulate the Krebs cycle, but it's like going into a little bit too much. Just know that they have to be reacted together and they form citrate, which is the next product. The citrate is then converted into isocitrate. Main thing about this step I want to point out is that this step is very non-spontaneous, so it's very unfavorable. So if you put this step in like in a test tube by itself, it would never happen. On its own, it's very non-reactive, and it's very unfavorable for this to happen thermodynamically. But because it's immediately used up in the following steps, which are more spontaneous than this is not spontaneous, it actually like balances out. So if the next step is very delta G negative, this step is a little bit delta G positive, but it'll still go forward just because it wants to go down that kind of like valley of just lowering energy. Okay. So that kind of pushes it forward. And that's why this step happens. That's the only thing really special about this step that you should know. Next, isocitrate is converted to alpha ketoglutarate. This is the first energy producing step, and it produces NADH from NAD+. Okay. So that's the first time we produce energy. And remember again, because you had two pyruvates from the glycolysis, that means you also have two acetyl CoAs from pyruvate dehydrogenase. That means you actually, for each glucose that you used, you actually run the Krebs cycle twice. So even though one isocitrate goes into one ketoglutarate, that makes one NADH. Because you're running the cycle twice per glucose, you actually produce 2 NADH from this step per glucose. Okay. Next, alpha ketoglutarate is oxidized to succinyl CoA. The enzyme used for this is very similar to pyruvate dehydrogenase in form and function. Again, because I didn't really cover the form and function of that one, it's not really that crucial, but you know, they're very similar. Has the same E1, E2, E3 component. They all actually have this fairly similar mechanism. The E1, E2, and E3 components have very similar mechanisms to the pyruvate dehydrogenase. And it uses the same cofactors, such as CoA, NAD+, and like those other ones that you didn't have to remember. And it also makes NADH. The general rule, I think, is if it's a at least for the Krebs cycle, if it's a dehydrogenase, that means it's using NAD+ or FAD+ to make NADH or FADH. So if you see an enzyme that has dehydrogenase in its name, at least for the Krebs cycle, know that it's making NADH. Like that's like a general rule that I would say. If it says ketoglutarate is oxidized to succinyl CoA, again, something is getting oxidized. That means it's a reducing agent. It's going back to our lecture on that. It's a reducing agent. That means it has to reduce something else. The best thing to reduce is NAD+ into NADH. When you do that, you make energy essentially by reducing that. Okay, so anytime you see something is oxidized, something is reduced, when you dehydrogenase, that usually means it's an NADH producing step or an FADH producing step. Okay, so this is the second step. Again, this makes 1 per cycle, 2 per glucose. So now we're up to 4 NADH per glucose so far. Next, we want to convert succinyl CoA into succinate. This is an ATP or GTP producing step. This uses substrate level phosphorylation. So this means just takes directly from the substrate, which in this case is succinyl CoA, just takes the phosphate directly from there and puts it on an ADP or GDP to make ATP or GTP. Right, and that's called the substrate level phosphorylation. CoA is released as well and recycled. Okay, so now we're up to per cycle 2 ATP directly, 4 NADH. Okay, and then the final steps. Succinate is oxidized to fumarate. This process is actually part of the electron transport chain and ATP synthesis, which will be covered next week, like I mentioned, but it's also part of the Krebs cycle. So what that means is actually the enzyme that converts succinate to fumarate is inside the membrane of the mitochondria. Remember, this happens in mitochondria. It's actually embedded in the membrane of the mitochondria, and that's why it is both in the electron transport chain, which happens at the membrane of the mitochondria, so it's partially in, partially out, and it's also part of this Krebs cycle. Okay, it involves FAD+ as a cofactor, which is reduced to FADH2, which is equivalent to, again, there's some debate. 1.5 ATP is like the newer value. Some say it's 2, but I think the MCAT uses 1.5. But I think they'll mention it between 1.5 and 2. Okay. And usually this FADH is directly used to send into electron transport chain, like I mentioned, and ATP synthesis. Okay. The next step, step 7, fumarate is hydrolyzed to malate. There's nothing particularly special about this reaction. Obviously, it's important, and obviously there's like, so if you were to do a very detailed dive, there's like an important mechanism behind it, but in terms of like overall, there's not really anything particularly important about it. So that's why I didn't put anything. Last step, and this is the step to go back to the beginning, oxidation of malate into oxaloacetate. This process also reduces NAD+ to NADH. This is the final step. Currently, after this step, we're on 6 NADH, 2 FADH2, and 2 ATP. Okay, just remember that. It also regenerates oxalic acetate, so the new cycle can begin once acetyl CoA is brought in again. Until it's brought in, nothing happens. It's kind of dormant. Then when you bring acetyl CoA in, the process starts again. This last step is actually very thermodynamically unfavored. So that means the delta G is significantly positive. But like the body does a special thing where it keeps very low concentration of oxaloacetate and manipulates the concentrations in the cell, which drive the reaction forward regardless. Okay. So the final product of everything, right? Adding everything together, we have 3 NADH, 1 ATP, and 1 FADH produced per Krebs cycle. Remember, 2 acetyl CoA per glucose, so double that. 6, 2, and 2. Add that to 2 NADH and 2 ATP produced in glycolysis, and the 2 NADH produced in pyruvate dehydrogenation. Adding this all up, you get 4 ATP directly, 10 NADH, and 2 FADH produced. With the electron transport chain converting the NADH and FADH into ATP, that is 32 or 36 equivalents of ATP. Again, depends on the source. There's some contradiction, but I think the newer value is 32 rather than 36. So that's what I would suggest probably going with, but 36 is also an accepted value, like a little bit older, but depends what the MCAT wants to do, but I doubt they'll ask you for counting. Okay, they'll usually tell you. So you get 32 or 36 equivalents per glucose used. That's significantly more than if you just got the 2 and the 2 NADH from glycolysis, right? And you didn't use that many new products, right? Because you look at this, you didn't actually really add anything or remove anything. You just put in the acetyl CoA, which again, you made out of the glucose that you already have, or the pyruvate that you already have. So you have this, and then you add it in, and this initially you had oxaloacetate, but then you go through this cycle, and you end up exactly with oxaloacetate. So whatever you started with, you ended with, right? You just use up this acetyl CoA, which is kind of like the remnant of the pyruvate that you had at the end of glycolysis. You didn't really have to use up that much more ingredients, I guess. And the CoA gets recycled in the end, right? Because the CoA is not in the product. So the CoA actually gets recycled. So you really didn't use up almost anything, right? Does that make sense to everybody? Okay. So that's about everything I wanted to cover. After this, I just wanted to maybe ask a couple of questions in the chat rather than poll questions, just some thought questions. But I know this was like a lot of information, a lot of memorizing that you probably will have to kind of like look over again. I would highly suggest it. If you are interested in learning about the enzymes, I would suggest like doing some research on your own and finding like the enzymes responsible for each step. They may ask you on the MCAT, not super likely, but they may, just in case, if you want to be prepared for anything. Yeah,